excited about this, getting to spend some time with Joni Rutter. So I was thinking about, Joni and I had the opportunity to spend a little bit of time this morning together and talking about a number of things, and I was realizing that I would love to hear, so we talked a lot about NCATS, which we will talk about, but I wanted to know about your background and your career journey, like how did you get to the NIH and, you know, kind of before you took on the current role that you have?
That's a long story.
We have an hour.
Settle in, okay? I'll tell the short version. Well, first of all, I just want to say thank you to you, Charlene, and to Nicole Boyce for inviting me to be here today and speak to you. I don't often get to speak to this audience, and I am super thrilled to be here. So thank you so much for the invitation. So my career journey really started, I'm originally from Kansas, and my mother's name is Dorothy. And so everything was a little bit fairy tale. I had an Auntie Em, I had a dog named Toto, it was the whole deal. And in high school, I for whatever reason, science really spoke to me. And so I really kind of caught the bug in high school that I thought I wanted to be a scientist, but I thought that meant that I had to be a doctor. I thought that's what all that there was out there. And so I spent the next several years of education trying to figure out how to become a doctor. And when it finally came down to it, though, I took, after college, I took a break and did a research stint at the Massachusetts General Hospital at Harvard Medical School and worked for Jim Gasella at the neurogenetics unit and worked on diseases like Huntington's disease, neurofibromatosis type 1, neurofibromatosis type 2, tuberous sclerosis, dystonias, dysautonomias, so a variety of rare diseases. And I thought that was fascinating, and it made me want to be a geneticist. And so I went to school to get a PhD in genetics, but they didn't have a program in genetics back then. I'm old, I guess. So my degree is in pharmacology and toxicology, but I studied human genetics for much of my career. And so that's really how I got to my degree. And from there, after my graduate work, I decided to do a postdoc at the National Institutes of Health. And for some reason, I did not leave. I did my postdoc at the National Cancer Institute and studied a variety of cancers, Mendelian cancers primarily, melanomas, BRCA1, BRCA2. And then after that, I decided to stay at the NIH, and I worked in the National Institute on Drug Abuse, because there was a fascinating intersection of how genes intersected with the environment that I was very interested in. And it turns out that there is nature and nurture, and both are very important for commonly inherited conditions. And so that was a really wonderful time. And then after that, because I was a geneticist, I spent a lot of time doing policy work at the NIH and ultimately went to the All of Us research program and became the scientific programs director there and stood up the program. And then a few years later, I got kind of tapped on the shoulder and said, hey, why don't you come over to NCATS? And I said, that's a brilliant idea. And I was lucky enough to work with Chris Austin for a little bit, and then he moved on, and I was lucky enough then to be appointed in the director role. So that's how I got here.
Well, we're glad that you didn't leave the NIH.
Thank you.
And so we've just talked about NCATS a couple times, and I expect that not everybody in the audience knows what NCATS is. And so maybe you could talk about what the center does.
Sure.
And also what the center does, and I think when you explain it, talking about what translational means would be also helpful.
Fantastic. So at the NIH, we have 27 different institutes and centers. And those institutes and centers are there to really direct the research across the country in various different areas. So I mentioned the National Cancer Institute earlier. So there's the National Cancer Institute, there's National Institute on Neurology and Neurological Diseases and Stroke, National Institute on Mental Health. So there's a variety of different institutes and centers that are there to study very specific organs, organ systems, or diseases. And there are a few, though, at NIH, a few of those 27 that are non-disease focused. And NCATS, the National Center for Advancing Translational Sciences, is one of those that's not focused on a specific disease. In fact, we're focused on the over 10,000 diseases out there, and most of those 10,000 diseases are rare, as you know. And so we have a sweet spot for rare diseases at NCATS, but it's not the only thing that we study. And so translational sciences, NIH is also known for the work that it does in basic and foundational biomedical research. It is the world premier organization to study biomedical research. And for about maybe, I guess it's now almost 12 years ago, there was discussion about, well, you know, NIH has been studying basic and foundational sciences for a long time. We really need to be thinking about translating that work and moving that into more clinical applicability. Now, most of the institutes and centers actually do translational research, so they've been doing it, but there wasn't anybody there that was really focused on how to do it better, faster, and cheaper, more effectively, more efficiently. And that's what translational science is all about. It's about understanding the science of how we translate things into the clinic. And so our mission at NCATS is to essentially turn research observations into health solutions. And we do that by understanding what the barriers are in moving something from the basic or the bench to the bedside. And if we can tackle what those barriers are, then we can perhaps make those move forward more effectively and more efficiently. And the reason that that's important is because we have over 10,000 diseases. We need to do things that are faster and better. It takes now about 10 to 15 years to get something into humans. And from there, it may take another 10 to 15 years to get it into the medicine cabinet. And it's at a very high cost of about a little over $2.6 billion to do that. And then lastly, the most dire number, I think, is that there's a 90% failure rate to get there. And so we're really thinking about how do we move the needle on those numbers to make them better. I'd really like to tackle that 90% failure rate. And if we can use translational science to get at not just the scientific barriers that are there, sometimes those are the most tractable, but those operational barriers, those administrative barriers, those financial barriers, those are the things also in our purview that we're trying to tackle as well, to help the failure rate be better, help us be better at predicting what might work in clinical trials. And so these are the kinds of things that we're working on at NCATS.
That's great. And I was really excited to read the strategic plan, your new strategic plan. I came from the tech industry, and so we often talk about the BHAG, the big, hairy, audacious goal, and I felt like this plan is the big, hairy, audacious goal. So you tell us a little bit about it?
BHAG, I haven't heard that. I'm going to use it. Yeah, so thank you. Our strategic plan is not out yet, and in fact, I have a call to action for all of you. We are putting out a request for information, probably next week, in fact, and it will be talking about the strategic plan that we're developing, and I would really love for everyone in this room to, if I could get your email, no, don't do that. Somehow we'll figure out how to get you the RFI, so that you can chime in and tell us what you think is most important for NCATS to be considering that might be most helpful for you. And that's what we're looking to do in building our next strategic plan, because our big, hairy, audacious goals, our BHAGs, are three. We want to bring more treatments for all people more quickly. And those are big and audacious goals. Right now, I don't know if I mentioned it, but you've probably heard it many times over, but there are about 5% of therapies for the diseases out there that we care about. And that number needs to change. It's been at 5% for a long time. And I think that we have tools, technologies, we have people in this room, we have superpowers here that we can start to applying on how we can start to move that needle. And I'd like to see that number go up to 25%. And I realize that that is big and hairy and audacious. But I would rather fail at 25% than succeed at 6%. Because if we fail, we're going to fail forward and learn a lot about that. And I think that's just as important. And so that's why that's a big goal. Now, for all people, this is not just about training and educating and career development for researchers out there, but this is also about building a real ecosystem that cares about health equity and diversity and inclusion in the research that we do, and making sure that our work generalizes to as many people as possible. And so that's the for all people goal. And then more quickly, one of the biggest examples I use for this is in the rare disease space, because we know that it takes a long time for rare disease individuals to be diagnosed. It can take 7 to 10 years. It took my mother 15 years. Some people never get diagnosed. And so if we can get the ability to move that diagnostic needle 2 times to twice as fast, that would be a big improvement. And even that's not enough, but that is an improvement that we need to see. So these are some of the things that we have in our 3 audacious goals that I'd love for you to chime in on and tell me what you think is most important for you.
Yeah, well, we can definitely disseminate that, to the attendees as one step from this conference. You mentioned your mom, and I don't know if you're willing to share a little bit about her story?
I would love to share her story. Thank you for asking. Dorothy from Kansas. My mother, when I first came to the NIH, which was a long time ago, 2003.
When you were 10.
Yeah, when I was 10. When I was Sadie's age. No, I think I was a little older than Sadie. She was struggling with something when nobody knew what, but she was tired all the time. She had bruises on her and couldn't figure out what was going on. But I kept saying to her, "Mom, there's something not right here." But I was in Maryland. She was in Kansas, and so it was very difficult. And it's a small town in Kansas. There's not a lot. It's rural. There's not a lot of information there about diseases. There are a few doctors that come in locally, and that's helpful. So over time, it took about 15 years for her to finally get a diagnosis, and she had primary myelofibrosis. Now, this is one of those rare diseases that's not a single Mendelian disorder. It's a rare cancer, and it's essentially something that where your bone marrow just it doesn't work right, and it starts to spit out these red blood cells that gum up the works in your bone marrow, and over time you just stop making red blood cells. But at the time I was at NIH, and so my foray into advocacy was really just that single experience of trying to understand what in the world is this disease, and how do I find out who knows about it? And so I combed through PubMed, which was the major resource that I had. And I found the one doctor in the US that was an expert in this disease, and he was amazing at the Mayo Clinic. He had a humanity about him. He had an artful approach to the science of this disease, and was really wonderful. That said, there were no treatments for this disease, and so we just had to take care of her when her symptoms would appear. And then over time, the main treatments were basically blood transfusions. And over time, they just stopped working. And so there came a day when my mother said to me, I'm done. And I've had a good life, and I've lived long enough, and I'm gonna stop. And that was a tough moment. And about a month later, I lost my mom. But as I'm here today at this meeting, it has brought sort of all of that back, and I think to myself, boy, if I would have had access to a lot of the resources that we've been talking about, the people that are in this room, understanding what you've been through and learning from that, that I think would have been very empowering. Wouldn't have changed anything, I think, for my mom for a variety of reasons, but I think that it would have been extraordinarily empowering to know that there were others out there struggling and having similar kinds of things. And so when I come to this meeting, it's my first time, but when I've come to this meeting now, it shows me that we're dealing with rare diseases, but they all have common issues, common needs. And we have to work together to try to figure out what those common needs are in a very kind of NCATSian way. These are those barriers that we can try to fight together and figure out how to make this move forward.
Joni, that is an eerie story. My mother had myelofibrosis, and it progressed to multiple myeloma after many years. We were kind of in that watchful waiting that happens with a lot of, sorry. I'm sorry about your mom, but this is, it is really interesting. I didn't, when my mother was ill, I knew it was rare, but this was, many years ago, and so the whole thing of kind of understanding that there was something like advocacy existed. I didn't understand that, I'm so grateful that this community exists. But how eerie!
That is. Yeah. That is amazing. We need to
yes,
talk more.
we definitely need to. Well, you talked about 10,000 diseases and 10,000 rare diseases, and this do things faster, cheaper, better, and that sounds like platform approaches. And so, what is NCATS doing in terms of approaches that are in this realm? In this realm,
right? You're right. We need platforms. When you're talking about 10,000 diseases, if we went one at a time, we'd be here for a long time. We don't have that kind of time. And so these platform approaches are really there you don't focus on any specific diseases, although we do, because we learn from very specific diseases and how we need to move those across the translational pipeline. But we take those learnings and figure out how we need to learn from those and apply them to the next. And you do that enough times, then you can figure out, we can actually build kind of a platform for this that can actually work on a variety of different diseases. And at NCATS, we support a variety of different platforms, and they come in different flavors. In the preclinical side, we have platforms for high-throughput screening of compounds. We have a library of 370,000 compounds at our disposal, and about 3,000 of those are FDA-approved compounds. And so if we get a hit with an FDA-approved compound, we already know a lot of information about that compound, especially in the safety area. And so we just need to do a little bit more in understanding perhaps the efficacy aspects of it. So you can talk about getting to more quickly, that's one way to do that. In another platform that we're trying to build for this idea of the 90% failure rate is addressing the sort of long-standing gold standard of using animal models or rodent models to predict what perhaps a human might have in terms of a disease condition, or 2D cell models, and that's not working well. It's not very predictive. It turns out that 60% of the time, these compounds that pass muster in rodents or 2D cells, they fail in phase 2 clinical trials, or 30% of the time they fail in phase 1 clinical trials. So we need to do something more predictive. And so part of what we're trying to do now is think about how can we leverage more human cell-based models to be those predictive engines for identifying how something might be predictive for a particular disease or phenotype. And so we're starting to build that kind of a platform on a variety of different diseases or organ systems that we can then compare perhaps a healthy state to a disease state and understand what compounds might work best for those, how to perhaps identify new biomarkers that haven't been identified before, or even just studying how the disease progresses in those. And I'll just say one fun fact is that as we built that platform, we have established a really wonderful partnership with NASA. And we've created these tissue chips, and we've put these primary cells or induced pluripotent stem cells from different organs, kidneys, heart, lung, liver, vascular cells, joints. We've recapitulated joints, blood-brain barriers, and we've sent them up into space because it turns out that when astronauts go up into space and they spend some time there, they actually have these aging-related problems. They get heart problems, they get kidney problems, they get muscle weakness, all these sorts of things happen. Now, when they come back to Earth, though, it reverts back to normal, which is a good thing. But we said, hey, if we could send cells up into space, we might get a chance to study those aging-related conditions a little bit more. And that's exactly what we're finding out, is that you can see in heart cells, just 3 days in space, you can start to see inflammatory responses and those aging factors happening. And so it gives us a really fast way to look at those processes and then figure out what's going on there and bringing that back down to Earth and applying that to other diseases and conditions. So that's how this platform technology is really effective. And then most recently, about a week ago, we just had a workshop on how to use these sorts of platforms, these tissue chips and 3D cell systems for rare diseases. How can we create a platform for these models specifically for rare diseases? And so I hope that we'll be able to generate something that will be very powerful for the community, again, to really address that more predictive need that we all have in moving therapies forward.
So following up on your fun fact, how is this working? Like, the cells go up into space. Are there do scientists also go up to like the space station, or how does that work?
So that's a great question. So we partner with NASA. NASA, we create these experiments so they're essentially pilot proof, because we can't send researchers up into space. But the pilots there, or the astronauts that are going up, they can work with the cells, and essentially we've worked with them actually to engineer the experiments, so that basically they just have to change the media or add a few compounds here and there. But at the same time they're doing that, on Earth, there's the scientists, the researchers conducting the same exact experiments on Earth. So we're comparing the Earth gravity field to the low Earth orbit or the microgravity environment. And so when they, about a month later, 2 months later, they send the cells back in those little pods that you've probably seen that splash down into the ocean. We grab those and we take those back to the lab and study them together. And that's how we know we're finding these inflammatory markers that are really prevalent in some of these aging-related problems.
Very cool, very cool. Well, you talked about a few of the platform projects that y'all are working on. I've also heard of the bespoke gene therapy program, and so can you talk a little bit about that?
Absolutely. So I was talking about more the preclinical side, and so now we're translating into the clinical side, and I think this is super exciting. So the Bespoke Gene Therapy Consortium is it's a public-private partnership with the foundation of the NIH, who's really leading this. And we're working with about 30 different partners. 10 of those are at the NIH, and about 10, there might be 11. I actually can't remember at the moment. And then about 20 or so partners, patient advocacy groups. We have a variety of pharma companies and biotech companies also who are involved in the Bespoke Gene Therapy Consortium. And what we're trying to do here is really thinking about this key feature of rare diseases, which is that there are about 80% of rare diseases have a single gene nature to them, a single gene disorder. So there's a mutation in a gene that's causing those disorders. So that's the link, that's the hook. And how can we then use the genetics to understand how we can then build a therapeutic gene and put it into a delivery vehicle to replace the mutated gene? And we can do that through viruses, adeno-associated viruses. We're talking about viruses during a pandemic, I will say. But of course, you know that these viruses are relatively benign. They have very little sort of inflammatory components or immunogenicity, I guess I would say. And so they're relatively safe. They've been used before. There's precedent for them. So we want to figure out how to leverage building a platform for those kinds of delivery vehicles, those viruses. And they come in different flavors, AAV1 through AAV9, 10, 12. Very, very creative in how we number them. But each of those flavors can target a specific end organ. And so it depends on what disease you're trying to affect, where those cells are, and how to use those particular vehicles to get them to the right place. And so that's an important feature for the adeno-associated viruses. And the Bespoke Gene Therapy Consortium is really trying to figure out how do we maximize and leverage that kind of vehicle. Right now, if you look at gene therapies, it's very difficult to get those therapeutic genes into the viral capsids. And so we need to do more biology to understand how to actually make that more robust. Right now, the percentage of the therapeutic genes that gets into those viral capsids is quite low. So we'd like to see almost a logarithmic increase in how those genes can get into the viruses. So we're not just sending in and treating patients with empty capsids or empty vectors, but actually making sure that as much as possible has that therapeutic gene in them. So that's one part of the Bespoke Gene Therapy Consortium. The other part is then understanding how the biology of delivery, getting it to the right cells, and so we have a variety of programs in those areas. But I think a key feature of the Bespoke Gene Therapy Consortium is that we are working hand in hand with the FDA. We can't do this without the FDA. And that sort of collaboration, that partnership really it helps us understand what are the regulatory needs so that we can build a platform that others can be able to use. And understanding sort of, again, how to bring those therapeutic genes into the capsids, how to deliver them most effectively, but how to manufacture those. That's where the regulatory components come in. We need to make sure we understand those sort of critical quality attributes of how to manufacture these kinds of gene therapies so that they can be as consistent as possible. And so the regulatory aspects are really critical for this. And essentially, the key word that I would say throughout this is really understanding how we can streamline the process of gene therapy. And I think that's the big thing that BGTC is doing. Now, I'll say that we have identified 8 rare diseases that will be going through the process of sort of testing this as a model. And we will be starting that over the next several years in terms of moving those gene therapies through those specific rare diseases to test this. And I hope to come back perhaps in a few years and tell you how that's going. But I think it's very exciting because even though this is a gene therapy platform, I think that it's sort of a platform in a platform. Because what we learn in this gene therapy platform and developing this delivery system, if you will, of getting therapeutic genes to people, that can translate very well perhaps to gene editing kinds of approaches or antisense oligonucleotide kinds of approaches or other types of modalities that we need to deliver in certain ways like this. And I imagine too that we're not gonna really end up here in 10 years in using adeno-associated viral vectors. We're probably gonna advance quite a lot in using mRNA technology, synthetic vector technology, and those sorts of things. So I think that there's going to be a lot of growth in these fields to help us even push forward on.
That's great. It is very exciting, and I think that I am coming from the software world, like the whole concept of a platform and building on platforms and utilizing them to make something basically totally pervasive is kind of critical, and it's exciting to see the biology side of that. I want to switch gears, and we've been talking a lot about science and and the technology for developing therapies. How how do advocates engage with NCATS?
Yes, great question. Well, actually, I'd like to hear from you all how you'd like to engage with NCATS too. But I think there are a couple of ways. One is that we have a website, and we have a variety of things on our website that we hope are valuable and usable for the rare disease patients and patient advocates. And I have a slide actually that I can show you here in a moment on that. So that's one way. I have a stakeholder listserv that I communicate with a variety of stakeholders almost monthly, maybe every other month. I'm not actually sure how often I send out those emails. But I know that several of you in this room are signed up for that listserv, and it'd be great if you're interested, you can go to our website and sign up for that email. But it tells you a little bit more about what we're doing at NCATS, what are the new initiatives that are coming out, what we're thinking about, et cetera, and also some resources that we have. So that's another way. I think another area where patient advocates can interact with us is through our Rare Disease Clinical Research Network program. Many of you are probably familiar with that program, and I think Tina Irv is probably here in the audience somewhere. And there's Eric. Hi, Tina and Eric. Tina Irv is the program officer for the RDCRN and is amazing. And we have 20 different Rare Disease Clinical Research Network, RDCRN consortia, and they study at least 3 rare diseases at a time for any given consortia. I think we study over 100 different rare diseases in that. And in addition to that specific consortia, there is a requirement to get money for this. There is a requirement that you have to be working with a patient advocacy group. And that group has grown over time. And I think that's another way in which this community can get involved with perhaps getting in touch with our patient advocacy groups within the RDCRN. They're great mentors, and they're going through very similar kinds of things that you're thinking about. And so that might be another way to engage. I guess that's not with NCATS, but it's with kind of indirectly. And then perhaps the last thing I would say is that we have a council meeting 3 times a year where we have advisors come meet with us, and we tell them kind of the different activities that we're doing. And they give us input and advice on those directions and some concepts we're thinking about. We have some council members in the room. And that's really important. But I think what's really unique with NCATS is that we have to have rare disease advocates and even rare disease patients on our board. And so there are many of you out here, and if you're ever interested in thinking about doing something like that, I'd love to hear from you. We can only have a certain size of our advisory boards, so there might be a wait, but I would love to see if anybody is interested in doing that.
Awesome. Well, that's great. So you talked about consortia, and that implies collaboration, and that's one of the key themes of our conference. And I am curious if you have advice for patient advocates, because a lot of folks are starting out, on their advocacy journey and just starting to engage researchers. What advice do you have in terms of building collaborative relationships or collaborative networks with researchers? If we could change our C in NCATS, that's what we should change it to, is collaborative. I think it's hard to say if there's a one-size-fits-all approach here, but I think the more you kind of find out about the things that you're interested in, finding those groups who may have been there before, and reaching out for advice or even collaborating on something, following along and understanding what they're doing. That's one way. But another way, though, is to get more involved in the research. We talk about level up on advocacy, which I think is a really cool way of thinking about it. But as I've been thinking about this meeting and what I've learned at this meeting, I also feel like leveling up the advocacy by design will also level up the science.
And so we need your help, I think, in some of these areas. And so a lot of times I feel like we were talking earlier, a lot of times I feel like that collaboration with a patient or patient advocacy group with a researcher is kind of a dotted line. And I'd like to see more of a solid line connection for those things to happen. So this might be a spot where I could show that slide. Can I do that? So it's not the prettiest slide. I think I made it myself. And what I do is I tend to use QR codes because that just takes you right to the website with a lot of information. So I'm going to give you the Cliff's Notes version. I don't know, do Cliff Notes still exist? I'm going to give you the Cliff's Notes version. So GARD, and Eric Sid back there is the GARD aficionado. GARD is a website where you can find out about rare diseases and perhaps the number of people with those rare diseases, what kinds of treatments might be available, if there are any clinical trials that are ongoing, who's doing research in those rare diseases. So it's a really great place to learn a little bit more. But also, GARD, I think, it serves a critical function because it has a 1-800 number that you can call if you're new to rare diseases. Maybe you're a parent who you really don't know what's going on, or you just your child just got diagnosed, or you just got diagnosed. There's a call center whom you can call and talk to about those specific issues, and they can help you kind of get started on understanding and navigating that process. So GARD is a great website. It's the top 10, I believe, one of the top 10 websites at the NIH, period. So it is frequently visited, and it just got maybe top 10, top 20, I don't know. Eric is going, eh. But it is frequently visited, and we've just modernized it in a way, and so I think you'll find it a little bit more user-friendly if you've been there before. It still needs more work, and we'd love to hear your thoughts on that too. The second resource is a toolkit for patient-focused therapy development, and I know that that's top of mind for many of you. And so this particular toolkit is really about giving you information on what you might need to do or be thinking about in natural history studies, how to be thinking about talking with the FDA, what kinds of things to expect. It also gives you a sense of how to go about finding researchers who may be studying something that's important for your disease that you're interested in. And it gives you the tools to find those researchers. At NIH, we have a whole database of everybody who is supported by NIH funding. And within that database, you can search on keywords. And so that database, there's a search function in that toolkit that allows you to find particular researchers who are doing that. And so I would encourage our group here to reach out, perhaps through those resources to find researchers who may be able to be helpful. And then lastly, we have RADAR, which is a rare diseases registry program. And this is really for patient groups and scientists who are thinking about developing a patient registry. It talks about what other registries are out there, how to get to them, and also what are the best practices in starting a registry, how to think about data collection and sample collection. And this is also something that's very important for RareX and Global Genes too. And so these are resources. I think we might even link out to Global Genes for some of these resources. So we don't just rely on our resources. We want to make sure that there are other resources out there that we can leverage as well, and you can take a look at what you need most. And then that last one, NCATS Rare Diseases Community Resources. This is sort of a one-stop shop for all of those pieces of information I just mentioned and a few other tidbits too. So I hope that this is something that is can be tangible for you and serve as at least initial resource to help you get started.
Thank you. So we're going to open it up to questions in a couple minutes. I have a couple more questions, Joni. So I wanted to circle back around to the point that you made around the dotted line.
Yeah.
And so what do you mean by that? And I think that, not everybody, I can't see Chris Hempel. Oh, she is there. And, for those of you who were in the opening keynote, Chris said, she would write these emails, she would sign them as Chris, people, the researchers would respond, they'd say, Dr. Hempel, blah, blah, blah. But, not everybody feels comfortable doing that necessarily, and what do you see that patients bring, advocates bring to the table where they can help to bridge that divide and that dotted line?
Yeah, it's a great question, and here again, it's probably going to be different depending on where you are and what sort of the status is of what you're thinking about for your disease. But I think that dotted line to me is where it can become more solid is really the people who are patient advocates and people who are patients, they're the subject matter experts. And so the research that you have conducted on your specific rare disease, the research I conducted for myelofibrosis was deep. And you have ideas, and sometimes the research programs that we have follow sort of a pattern, and we need new and fresh ideas. And so I think it is definitely worth sending those emails to researchers to say, hey, I'm a patient advocate. I found this paper. This sounds like it's really important, and I see that you might be working on that. How can we help one another? I think that's the start, and really I think advocacy, what I do and how I approach things is really about relationship building. And I think if you can build relationships in that way, you start to generate that trust. But I also think that there is this idea that in software development, product development, there's this thing called user experience and user design. And the iPhone 15 is coming out, and it's got this new port, the USB-C port. I don't know if you've heard about this. That's a big deal, but I bet you dollars to donuts that there were a lot of people who came to Apple and said, hey, we want a USB-C port. Otherwise, they wouldn't have done it. And so it's understanding what are the needs of the community and how do we help meet those needs. And I think that user design approach that we've learned from software development and product development can be applied just as equally to the translational science that needs to get conducted with the partnerships of rare patient advocates and rare patients with researchers. And so that's where I see promise and hope, and why I'm excited to be here at this meeting too.
Yeah, I actually, to that end, I can share an example of that from my daughter's condition. So we were very interested in developing a disease concept model, and because we felt that it was important to be able to document what were the significant symptoms for our patients and how it was impacting their lives. And this was several years ago, I approached one of our lead researchers, who's a clinician, about this, and he did not think that it was necessarily like strong science. It wasn't, like going and seeing patients in clinic and, gathering that type of data. And so it took a long time to convince him that this was a worthy effort and that it was important for engagement with the FDA. And now, so fast forward, I had to send a lot of papers and more explanations, but fast forward, 3 years, and we have a disease concept model. It was published late last year, and now he is a complete convert. He, in terms of the value of it, and in terms of being able to utilize it in other disease communities. And I think one of the key themes for us, because my daughter's condition is a neurodevelopmental disorder with a significant a high incidence of seizures, was that seizures are impactful, but they aren't the whole story of the patient, and that came out, in spades in the data that was collected. So I think it was just, for me, a really great experience of getting on the same page with a researcher, and then having a better outcome because we were able to collaborate. Us bringing the patient viewpoint and our researcher bringing the scientific rigor. Mm-hmm. So.
That's a great story. We need to have you and him sitting here someday so you can tell that story together.
Yeah, that would be, that's a great idea. So I have one more question for you before we open it up, and this is on repurposing, because there's a lot of interest in drug repurposing. And I've talked to many people just in the last day and a half around that they're starting projects, they're trying to start projects, and and you brought it up earlier in terms of this, a huge library that y'all have access to. How would you advise groups on starting to pursue repurposing projects, and what thinking should they have beyond just the technical, what's going to be
Does
their
it
top
work?
hits?
Yeah. This is a great question, and I feel like this landscape is going to change a lot. It's going to do this kind of thing. Drug repurposing there's promise there. We know it. We've seen it. We just went through probably this whole last 3 years of COVID has been an endless cycle of drug repurposing in a way, and I think we really have a good understanding of how to do it well. The issues, though, are not so much scientific as you kind of alluded to. They're actually more operational, financial, and even administrative, and legal as well. So policies around this area are policies and legal issues are tricky, because a lot of times if there is a drug that is marketed that might be something that could have a new indication for a rare disease, it is often that a drug company owns all the rights to that particular compound or drug, and they may not be interested in actually licensing that for a new indication, because if for some reason that medication doesn't do well or fails in a clinical trial, that could affect the bottom line for the indication that it's being used for. So there are real sort of bottom line issues around this. But at the same time, they might be interested in figuring out what those new indications are. We know that because it has happened. There are also drugs that are out there that are generic, and so have gone through their period of patent protection. But there's not a lot, it would cost a lot of money to then recycle that into doing a clinical trial. And that's a pretty weighty endeavor in terms of the costs associated with that, because you're not going to recoup them because it's already generic. So it's the economics of drug repurposing is part and parcel, I think, some of the things that we need to get more creative around. And I think we're trying to figure out how best to do that. Maybe we need to get into more of the preclinical stages while they're still doing their indication finding. Maybe that's a sweet spot. Or maybe we continue to try to really pursue this idea of looking at marketed drugs that are out there and finding those new indications, and just really working with the companies as best we can to see if we can move those forward. But I will say that here's an area where data science is leading the way. We're learning a lot through gathering a lot of data, putting it into we call it a biomedical data translator, because it has all of the sort of the drugs that we know out there, and perhaps what those indications are, and then PubMed resources to perhaps identify new indications that drug might be used for that we couldn't really identify just by looking through PubMed ourselves. We need AI to help us sort through that. So we've created a lot of work in this space to create this sort of resource to do this, and we're currently going through user experience and user design for this. So it's not ready for prime time, but I think these are the kinds of resources that could help us ultimately push these ideas forward, so hopefully we can get some more traction in drug repurposing.
Awesome, thank you. Well, I see that there's a couple people queuing actually for questions, and I think is that you, Harsha?
Yes, I am.
Go ahead.
Thank you so much. Dr. Joni Rutter, my name is Harsha Rajasimha. I am a former NIH genomics scientist, and also the founder of Indo-US Organization for Rare Disease, and the developer of a software platform for making clinical trials universally accessible, particularly for rare disease.
Awesome.
Thank you so much for your leadership at NIH and for sharing such insights today, and the resources, GARD and RARE are phenomenal, very valuable for the community, and we use them quite a bit. Over the years, have referred patients to GARD and have been referred patients back to us as well. My question is on the article that you kicked off this year in your blog for the Global Village article, that each rare disease is a global village, and that really resonated with us and the community. So my question to you is, what advice would you have for patient advocacy groups and the ecosystem at large on how to approach this global village, as most of the work still continues to be heavily focused in certain parts of the world, and especially emerging markets and the rest of the world. How do we engage communities there, especially for ultra rare disease where the numbers are so small in any one country?
Yeah.
And also, if you can say what NIH could do or is planning to do in that direction, but with its own limitations, what could, outside of NIH, what should happen? Thank you.
Whew, that's a great question. Thank you for asking that question. I am very interested in understanding how we can work more closely with international communities, kind of for the very reason you just mentioned. You might have one patient in different countries with a rare disease that's going to be important for us, and adding them into the sort of broader milieu of helping us advance what we know about that ultra rare disease is critical. So the more we can find these individuals, the better off we're going to be. So I think that understanding sort of how we can build networks to do that better is something I'm interested in. And one of the things I've been trying to do is actually work with our, since I'm in Washington, D.C., working with a lot of the embassies there to tell them more about what NCATS does, who we are and what we do, and our work in rare diseases for that very reason. And I'm hoping that there might be ways in which we can find partnerships with a variety of different countries and thinking about that. But they all have very, perhaps ideas that might be different, but where we can align those, that's where we want to find those strengths. And so I think that the more we talk, the more we build those relationships, the more powerful that can be. So I think it's a goal that we're having to start to build those relationships with those international communities. And you know, as you've alluded to, it's just critical to be thinking about getting to that clinical trial space. We need as many patients as we can possibly get. We can do that better with the international community. The last thing I'll say about that particular area is that in the RDCRN, we have grown that program to include more of the international communities. And the scholarly publications that have come out have just, they're just exponentially growing because of those relationships. And so I think that's another strength that we have in building those partnerships. So I think there's a lot of hope here, and it's really about finding where we can work together on that.
Aaron Aben with the Autoimmune Registry. Thank you. Regarding repurposing, there's a group called Cures Within Reach that everybody should know about. They run grants and things like that, so that's, I just want to put a plug in for them. One of the things about translation is that you're talking a lot about getting from the bench to the medicine cabinet or to the pharmacy. Getting the last mile is in the health system.
Yeah.
That final step.
Yeah.
So is NCATS ever going to get into what's called the learning health system? I'll leave it at
Okay. Thank you. It's a great question. I think this is one of those areas that is also really important for rare diseases, because one of the other issues with rare diseases, one of these big, hairy, audacious goals, BHAGs, is, I'm learning, is that you know that it's very difficult to find rare disease patients in the electronic health record system. Very hard. We've done an economic burden study. EveryLife has done an economic burden study. We came up to the same answer using 2 different approaches, but we studied 14 rare diseases for our study because we knew with confidence that we could find those 14. But how many rare diseases are there? A lot more than 14. So this is one of those bigger problems. And so I think in terms of a learning health system, we also have to kind of raise our game in terms of being able to find rare diseases in the electronic health record community. And that's an area I know that many people are very passionate about, interested in, including Eric Sid in the back. And so that's a problem that we're also working on. But I think what that can do then is the other major flagship program that NCATS supports is called the Clinical and Translational Science Award Program. And this is a program that supports 63 academic health organizations across the country. Some would say that it's a good foray into this learning health system environment. But what they can do is start really testing that. And one of the areas that I do see promise in is in electronic health records and understanding how we can use electronic health records to identify perhaps new diseases or identify or even predict someone who might have a rare disease before they go to see their 7th doctor. Maybe we can start to predict that when they see their first doctor. So I see real value in this idea. But not only that, we're using that sort of a platform in that learning health system model, we can start to also create clinical decision support tools for physicians who are seeing rare disease patients so that they might hear the hoofbeats, but maybe they don't even have to think about it anymore because they can rely on clinical decision support tools that are built in. So a lot of the work that's being conducted in artificial intelligence and machine learning are starting to get at these things, but there's a long way to go. And we have a lot more to learn. So I think that this is an area that has a lot of promise. I think we're seeing more of this idea of a learning health system approach, but I think we have yet to see really how it's going to take hold. But once it does, I think it will be highly valuable.
Great. Any other questions? So I have one parting question, which is, so we've talked about a lot tonight, and at the same time, going back to the early comments that you made, 5% of diseases have an approved therapy, and so it's still a long road for a lot of disorders. And so, what makes you optimistic, or hopefully you are optimistic?
I am.
If you are, what
am.
makes you optimistic about us being able to tackle all these diseases?
Yeah, it's hard to sort of pinpoint it on one. I'm an optimistic person by nature for some reason, and I think that what makes me optimistic is that we do have we have tools that we've never had before. We have electronic health record systems that we can use in a very strong way. We just have to figure out how to make sure we can do that. We have a large room of patient advocates. This is a lot of what we're talking about is not it's not a science problem, it's not an engineering problem, it's an ecosystem problem. And so the more we can build this ecosystem and network of people working together and providing the ways to facilitate those partnerships, that's what gives me hope. And I will say that, I started off talking about my mother because you asked, and now I want to share it with you because there was I think it was maybe Monday that it came out that there is now a treatment for myelofibrosis. And do you know what the treatment's name is? It's momelotinib. Mom is in the name. And so I'm really proud of, this is a GlaxoSmithKline just came out with this announcement, and I'm, so these are the kinds of things that make me optimistic.
That's wonderful. Well, Joni, thank you so much. Thank you so much.
Thank you very much, Charlene. I really appreciate it. Thank you all.